Good afternoon, guys. I'm Dr. Manoj Nair, aquaculture scientist for the COM Land Grant Program in Ponape. I work out of the Land Grant Central Office here in Colonia Town, and I'm in charge of the aquaculture program throughout the Micronesian region. For the time being in FSM, we are having a multi-site research project on sea cucumbers, mainly the Sandfish sea cucumber. It's a couple of sandfish we have been rearing. As you know, sea cucumbers are highly economical in their value, especially for the Asian markets, and that's the reason why we have chosen sea cucumber, sandfish sea cucumber, as one of our candidate species for aquaculture. The aim of the project is to provide economic development opportunities for the Micronesian people, train them, and through research and extension, we try to develop a technology where Micronesians can produce hundreds and thousands of sea cucumber for making money and selling to the Oriental market in Asia. So in that regard, this project started in 2014. Actually, the project started in 13. I came on board on 13, and 2013 the project started in 2014. So right where we are standing is the hatchery part of it in Ponape. Actually, this project is also simultaneously running in Yap State and also in Palau. We were supposed to do Marshall Islands, but unfortunately Marshall Islands doesn't have this particular species, so we might be trying another species there. So at this moment in Ponape, Net Point is a facility where we are doing the hatchery part of it, because this program is based not on wild collection and cutting or anything like that. We are doing artificial breeding and production of juveniles through biological methods, where we bring in the male and the female and spawn them and get the eggs fertilized and produce the juveniles. And the juveniles are used for farming research project. We have currently a site in Soques Municipal in Ponape, where we have a huge farm, 100 by 100 meters long, a hectare farm, where we release the juveniles out of our research programs are released there for studying the growth of this species. So right now, like I said before, we are standing in the hatchery section, but right now it's the grow out section. We'll move inside and I'll show you the hatchery steps. Actually, these guys have already given out 4 million eggs 2 weeks, almost 11 days ago, and we are rearing them in the tanks inside, and you can see the process and the stages of the larvae inside, and we can move inside. So this again is a sandfish sea cucumber, called in scientific terms called Holothuria scabra, and in Ponape it's called a langon. So let's go inside and start the first section of the tour, which is the hatchery part of the program. Thank you. Part of the project where we are hatching the eggs which we got from those animals you saw in the last segment, and we collect these eggs in sieves like this, and the eggs are fertilized and put in these tanks. And we have a stocking density, and according to the number of eggs we have, and then the size of the tank, we put them in these tanks. And every other day we collect the eggs or the larvae once they are hatched in sieves like this. This is a sieve cloth of different mesh size, because the larvae are of different sizes. So we drain out all the animals in these sieves, and we collect from these sieves into the tank. If you look in that side, you can see very, very small babies inside. So once we collect, we look at them under the microscope, and we count them, and then we put them back, or we put them in another tank. Why? Because this is a controlled environment. I did tell you, the whole place you see, the water inside is salt water, and the salt water is coming from the ocean outside. We are around 100 meters from here. We have a long pipeline. The water is pumped by the pump on the other side, and it's running through these green These contraptions, these are the filters. So the entire dirty water is being filtered to different sizes. You can see the filters out here. This is how a filter looks like. So the water goes through this and it's coming out at different sizes as the net goes. And finally there's a UV unit. So when water is filtered, the animals, gross animals and the eggs and those things which pass through them are still alive. So these can also compete with your animals inside. So we want to kill everything in the water. So we use the UV light to kill all the water which is coming past the filter. So basically the water is totally sterile. There is nothing in the water which is growing. And that is the water we pump into these tanks and fill them and put the larvae inside. So like you might have a question now, okay, how is the animal going to feed? Because you put all this animal inside and the water is clean, no food inside, right? So we have to produce food. So we have an algae section inside. Okay, when I talk about algae are minute microscopic plants from the ocean. So these are the plants which the sea cucumber larvae and the juvenile eat. So we have to grow them in the algae room in a controlled environment and mass produce them. And according to the number of larvae we have, like I said, when we count, we know how many larvae we have and how many animals we have. Accordingly, we give them a cocktail of microalgae. So I'll show you the microalgae room and we'll take the next step there. If you look inside, it almost looks like a hospital. It's totally controlled. There is aircon. You keep the temperature to 21 to 23 degrees Celsius. And like I said, these are plants. So what does a plant need? Plant needs substratum. Plant needs food. Plant needs light to produce carbohydrates through photosynthesis. So what we do in the same thing, we have cultures of microalgae. These are different kinds of microalgae. These are stock cultures. These are the cultures we produce in mass amounts. So this algae, like I said, this is sterile seawater. The seawater which we just pump through the filters and UV. We use them in this. Also, we sometimes even doubly sterilize by using the autoclave machine outside. So you autoclave this water, so water is totally sterile. Then you add to the water, you add chemicals just like the fertilizer, and you add the inoculum just like the small shoot of plant. You put a little bit of the stock culture of the microalgae which is growing in the ocean. So these are pure cultures. So it means only one type of microalgae is growing in this bottle, not so many of them. So we have different species of microalgae growing in different bottles. So from this bottle, you go to the next stage, which is you don't have it yet because we have already finished. You go to a 3 liter bigger bottle like these, and to a further bigger bottle like this, and from there we go into a thing like because we want lots of food, because as the animals grow they need more food. So we grow them in bottles like this. So if you see, there is a different change in the color. See, these are light, this is a little more yellow, and from there you are going into really dark. This is very densely filled with microalgae. So here again, this is air, because we need to aerate them, because there's no, because it's again like I said, it's controlled. Just like you're aerating and putting air for your broodstock outside or your larvae, you need to aerate them. You see the lights, because we mimic the condition of the ocean. 7 o'clock in the evening, we have a timer there which will turn off. Accordingly, at 7 p.m. this one will turn off and all the lights will go off. To mimic, this is night time. And morning, 6 o'clock in the morning, the timer will come on and all the lights will be on. So this is how we do it. Why we have kept it very low, even though you will say the temperature in the ocean is around 29 to 30 degrees. Why this is 23? Because we don't want it to proliferate very fast. Because we are wasting a lot of energy and money, we don't want it to run very fast and the whole system have to be run again. So we control the amount of algae we need, and accordingly we manipulate the temperature so that they don't grow very fast. So this is the algae section. Like you saw inside, they sieve the animals in the sieve, and from there they put them in a container. It could be a bottle or anything. Why? We need to know how many animals you have finally in the tank. So the only way to do is to take all the animals out, sieve them on a sieve, and put them in a bottle. From there you take samples, and you, in these little bottles, in amounts we know, like 1 or 2 ml per pipette sample, which you have seen before. And then we count all these animals in a chamber. So these are graduated. The chamber is graduated. You can see small graduations on the chamber, very, very minute. You can look under a microscope. And you put a ml of the sample water in it. Then you look under the microscope, take a little bit of formalin and kill those animals in the chamber, and you count all of them. So then you know in 1 ml so many animals are there. You extrapolate. So in 20 liter or 100 liter tank, so much, in 1 ton, so much. So we get 20,000, 100,000, 1 million, whatever it is. According to that count, you calculate today you need X number of cells of microalgae to feed one larvae. So we have so many larvae, we need so many liters of microalgae, because we also count the microalgae inside. This is called a hemocytometer. Actually, hemocytometer is usually used for counting blood. The same thing we use to count algae. Same principle. It has graduations, and you put 1 ml in it and count how many cells are there in the cells of microalgae are there in there. And accordingly, you know, okay, 1 ml, so much. So this culture of that bottle contains so many millions of cells in 1 milliliter. Okay, so much liter or ml, whatever, according to the day of growth and the number of animals and the concentration of algae, we calculate the food and give every day morning, and 1/3 of the food we give every day evening. So we feed them like 8 or 9 o'clock in the morning, then we feed them like evening 4 o'clock. So that is the process here area. So the swimming larvae is going everywhere and finding out a suitable place for them to go down. So by day 10 to day 15, within 2 weeks, they change the morphology from a flat swimming thing to slowly like a rounded sea cucumber. So if you see this picture here, this is like a ball now from this flat swimming sea cucumber. So once this is there, we go to, they are ready for, we call it settling. Settling means they are swimming. These are not swimming animals. This is not a fish. This is an animal which you see down in the ocean, down in the floor. So they are ready to move, transform themselves from a swimming animal to going down. So at that time we call it settling. So during settling, we put some plates, which I'll show you outside, and these swimming around animal, they go on the plates and they sit on the plates, and plates are also painted with food. And that food, they graze on that food because now they become like a sea cucumber. They have the tentacle and they are moving around and eating on them, and they start growing them. And once we can start visually seeing them, and around 0.1 to 2 gram, we take them out and goes to the next nursery, actual nursery phase. There are 2 nursery phases, one inside the hatchery building, and the second nursery phase is in the ocean. So for us, the second nursery phase happens at Nico Marine Park, where we have floating structures with nets. And the animals, they grow to around 20 gram size. Then it goes to the grow out, the final grow out to the farm, where we put them in the sand, because the basic morphology at the end of the day, they eat sand and the organic matter in the sand. That's how the sea cucumber lives. So it has to be in the ocean, in an area where there is sand. So by around 20 grams, because you put earlier, the problem also is there are a lot of predators which eat the little baby sea cucumbers. So We have found out through trial and error 20 grams is kind of good enough size, weight for them to survive in the ocean. So that's the size when we, from the nursery, second nursery, when we find animals in the floating structures or floating harpa cages of that size, we take those animals, weigh them again, make measurements and take our data and release them in our farm, where they grow around 1.5 years to be a big adult sea cucumber, the market size of which for this particular species is around 400 grams. So the aim of this is now still a pilot project research phase. The aim is, as we go along in the next 6-8 months to 1 year, mass production. Can we do mass? Because a businessman wants 10 ton. He doesn't want 5 sea cucumber. So the aim from the pilot stage to mass produce. Now we are trying to iron out all the problems we have in the hatchery and in the grow, social as well as biological. Once we start figuring out each problems and making it much better, the efficiency of the production, even in the world, you'll see there are, like I mentioned, we got 4 million eggs. That doesn't mean 4 million eggs, 4 million sea cucumbers will come out. The survival to juvenile is only 1 to 10%. That itself is very good all over the world. So our aim is to improve that. So we get more. Because if you look, this all is energy sucking. Yeah? You have aircon, you have pump, you have filter, all these take energy. Because the water has to be pushed in and so many things. So as much as you can produce in one cycle, you're more efficient. Just producing 20 animal or 50,000 animal, it takes the same effort. Because all these lights have to be on, all this pump has to be pumped. Pump cannot be pumped for half a tank or a full tank. It will run on its own. So that is something we are trying to improve as we go on. And the second phase of the thing is to get as much as Micronesian people trained in this program. So we are happy, Brian. We have a good relationship with COM-FSM, where we have a close relationship with Brian Lynch, especially marine science. So during summer, we get interns from the marine science program who come and work with us for a couple of months. And some of them who are interested, we always tell them, if you're interested for a short-term internship with us, we paid and we hired them. Like this boy here, and we have another boy in Yap, Owen. He's also a marine science graduate, but he's he's doing some work there. We have Elijah, who is also working there, also a marine science graduate. So our aim is to hire and train as much as possible Micronesians. That's the aim of the project. So Micronesians also know to do all these things. It's not very hard. They also are hands-on in this, and also do a business venture or something. You have opportunity to do business in aquaculture field. Like you see around 90% of what you see around is water. So you have to efficiently, sustainably use water to improve your resources, which are getting depleted through illegal fishing or through natural disasters. So that is the aim of the project. We are partners with usually with the local fisheries departments. Here in Pohnpei State, we partner with the Office of Fisheries and Aquaculture, or OFA. In Yap State, we are with the Yap Fisheries, Yap State Marine Fisheries. In Palau also, we work with the Palau Fisheries departments. So that's basically what we do here. So right now I'll show you the second part of it, what I was talking about, the plates and the nursery, and then we'll go to the. And now once we count the larvae which are inside today's larvae are 50% ready to settle, we'll count them and we'll pour the larvae in these tanks. So, and they'll slowly start going and attaching on these kinds of plates. And then we feed them microalgae, and as maybe a couple of weeks later, once we see them visually, we change the food. Here too, starting it's the really clear sterile filtered water, and as slowly as we see them attached and become bigger, we change to the raw ocean water, because ocean water already has food inside. and we put some additional food also inside. And here they grow for at least 1.5 to 2 or even sometimes even 3 months. At that time, by 2 to 2.5 months, once we see animals, big animals, which what our criteria is here, we have a hapa net outside. If it cannot go through the hapa net, we collect. Because we don't want to put them too much inside and feed and waste a lot of our resources. Once it goes in the ocean, they grow faster. So as we see animals of which are, we call it shooters, we collect them. So the other animals can also start growing faster because we are putting food inside. So that's what we do here. So 2 to 3 months here, and then it goes to a nursery section 2. So around 1 to 2 gram size animals, the ones Jackson was holding in his hand, that is a size, that is much bigger, but even smaller than we in Nico, which is a floating structure. I can, if you have time, you can come there, or I can give you some pictures, you can put those pictures. And those, there they grow for 6 to 8 months, and once they are my finger size, around 20 gram size, they are ready to go to our real farm, which we call the grow outs. So the whole thing has a hatchery phase, Nursery-1, Nursery-2, and the grow out. So what we have found through our previous experiments is it takes around just before 2 years to reach market size 400 gram animals from the egg. The day of spawning, it takes within 2 years to reach market size. So that's what we have found. This is a monotonous job. You should have a passion for it. It's not easy because everything is labor intensive, because you have to drain the tank. You cannot just train it very fast because the animals, the larvae are very fragile. So it takes a lot of time, energy. We have a common funny saying here that every day is Monday. So we work 365 days for this project. So we are working for us, and they are well trained, and like I said inside, our aim is to train more and more people and get people more aware of this way of sustainably doing through hatchery propagation, produce more sea cucumbers for economic development or for farming them, trying to just collect all your sea cucumbers from the wild. So we also sometimes release animals in the MPAs, our excess animals we also release in MPA, so there is also a production of animals for the wild also for people to see that there is conservation efforts also going from the land grant program. And if you have any questions or comments, feel free to email me or call me at the office. I'll be more than glad to help you out with those. Thank you.